Okay. Okay, so we will go and speak today about the about methodologies that we could find in brain research, especially regarding. A second. Do you see my slides?
Yeah.
Yeah. Okay, especially regarding high cognitive functions. So here I listed a few high cognitive functions, and these functions are. I mean, you could probably think of more, or a few seem strange. Why would there be high cognitive functions? But in any case, this is just a representative list. And what we will ask today, our central questions would be, and we'll do this through many examples, which I'll bring. The questions will be, first of all, how do scientists map these high cognitive functions to brain activity? Okay, we have the functions on the top, and we have brain activity, and we want to do some kind of connection between these two realms. Now, and another question which will be part of our discussion today is what types of scientific explanation can be articulated. I mean, let's say the question will be, are these explanations causal explanations or correlational, or maybe something else at all? I mean, we have to try to think what kind of science are we basically doing when we're doing the science of cognitive science. So let's start with some history. And so we won't just fall into the topic, but we'll be short. The short history is I'm showing you a list of people from 500 BC. By the way, BC is before Christ, not before Corona, so it's not. And what we see here is a list of philosophers and medical people which try to locate the place of the mind. So, and you see it alternates. Is it in the brain? Is it in the heart? Brain, heart, brain. And that is part of the riddle. And I'm using here the word mind as a general concept. Of course, and they didn't always define it this way. And to those who speak Hebrew, we have even from our Jewish sources, we have Shlomo asking "V'chochma me'ayin timatzeh" from the book of Mishlei, and here are two answers. Rabbi Eliezer omer barosh, Rabbi Yoshua omer balev. So you see the same options exist also here. So let's go forward in history, and we're getting to the 18th, 19th century with Gall, Franz Joseph Gall, which offered the idea of a phrenology, which is measurements of bumps on the skull to predict mental traits. So that was developed by him, and they had you could see in the next picture, you could see that all kinds of ways of measuring, and they even eventually built these big machinery to measure the bumps and to give you an output of what kind of mental traits do you have, and what kind of person you are. Of course, that in itself could be an interesting idea, but of course, as you all know, led to bad science and to bad results of this scientific possibility in way of in that they defined people as he's a criminality type person, and they have disease and healthy, and in that way to try to categorize people just according to these bumps. Okay, so but now let's go further into history, and now I'll really start to go into different types of studies. Now, the studies that I'm going to talk about today could be divided into two types. One is a type in which more perturbation studies, in which there has been some perturbation, either perturbation or some kind of I mean, the system was perturbed either by some lesion, like we'll see soon, or by intention, which we'll see in other studies. Or that's one type of a way to learn about the brain, and the other way about the mind and the brain, and the other type will be more observation. So you have in mind these two methods. One is more perturbation, and one is more observation. So let's start with the first type of lesion study. It goes back to the famous Phineas Gage, which I'm sure most of you heard of him. He was a railroad worker, and in an accident, he had a rod go into his medial prefrontal cortex. And as you could see in the picture, it was a very massive rod and did a massive lesion. However, he lived, and but what was realized is that he had a lot of social and emotional issues, and it wasn't well defined at the time. But for example, I'll bring an example now. Antonio Damasio was fascinated by his case and really tried to map people with these types of lesions in the prefrontal cortex, and to see what types of behavior or lack of behavior or different behavior could you see in these with these lesion patients. So this paper that he talks about damage to the prefrontal cortex increases utilitarian moral judgment. So let's look into this study for a moment. The study takes people and asks them, gives them a case study, and they have to decide in this case study what would they reply as the right moral judgment in this case, or what would they do in such a case. So these are famous cases. The first case is the trolley case on the left, in which a trolley, I'm sure again everyone knows this, but I'll just go over this fast. A trolley is going straight on the tracks, and there are 5 people on the tracks, in which it will kill if it continues that way. However, there's a way to diverge the trolley by a person that's disconnected and is standing on the side, and he can move the leverage so the trolley will diverge to a side track. However, in that case, he will kill one person. And the moral question is, do you let the trolley go on, go its course, or do you do you actively move the handle and it will kill one person? So that's the original articulation of the problem. There came later another way of presenting the problem, and that is the man on the bridge. That's the middle picture. It's the same story. The trolley is going straight and will kill the 5 people lying on the tracks. However, you're standing next to a big person on the bridge. If you push him down, he will fall. The trolley will kill him, but will save the 5 other people. So the consequence is the same consequence. You act and you push the person down, and then you save the 5 people. However, when people are presented with these 2 options, with these 2 stories, usually in the first case, they will say that that you should diverge the trolley and kill the one person and not the five, and that's what's called the utilitarian reply, because you count what's better, and the better good is that one person will die and not five, and here you do the act and one person dies and not the five. However, when people are presented with the bridge, here already the emotions come in, and people find it hard to push someone actively from the bridge to save the five people. And in this case, people usually reply that they would just let the trolley kill the five and not push the person. And now there's another version, which is even a higher conflict, and that's the crying baby version, which is people are hiding against the enemy. A group of people, stories that we are not familiar to all of us, are hiding against the enemy, and you have a crying baby, and now if you stop him, you shut his mouth, and he stops crying, you'll be saved, but he will die. And if you let him cry, then you'll all be caught, and everyone will die, including the baby. But here also, this is a very emotional situation in which people many times choose the emotional reply. However, when we check the patients with the lesions in the ventromedial prefrontal cortex. So these patients, in the case of the high conflict dilemmas, they give actually a more utilitarian judgment than the control subject. So that's the way you do these studies. You ask the control, the healthy subjects, how would they reply, and you rate it, and you ask the lesion patients how they would reply, and you rate that, and then you have the answer that they have something different here. Now, I'm just to emphasize one little point is that I'm not saying what is more moral and less moral in these cases. I don't know. We should think about it philosophically. But what they're interested here in these studies is to see do they react normally as would normal participants react to these judgments. So it's not saying anything about morality. Maybe this lesion made them more moral. I don't know. But in any case, you would find what changes. What's the change that exists here in these lesions? Okay, and if you want to look deeper into these, or look into a list of how human behavior changes from these lesions, here's a nice review paper from Bob Knight's lab. Okay, now that was the lesion patients, and now we have to let's think of what happens here scientifically. Okay, so first of all, these cases are often rare cases. Okay, and they're not something that you have, and happily enough, they're rare. I mean, they happen either as a result of a trauma or what is it called? Blood clots or things like that in the brain, or some kind of sickness. So that's how you have lesion patients, and the lesion area is usually not very focused. It could be contain not only one specific area, and also it's interesting to note that after a few years, these lesion patients compensate the lesion with brain plasticity, and sometimes they regain abilities which they didn't have in the beginning. However, you do have unique insights with these patients. You learn a lot, from them. And what we're interested in our question is it a causal? What do you learn causally? So yeah, you have here a causal role of the damaged brain region. You know that this brain region is playing a causal role in this behavior. However, interpretation of causal roles are very, you should be very careful about it. For example, let's say my battery, my phone is, the battery fell out of my phone. So you won't hear any music on the phone. But now is the battery the causal reason for music on the phone? In a way, yes, but on the other hand, it's a very distant causal connection. So so we should be aware of this causal connections that we do here. Any questions before we go on? Yeah.
Just one comment about lesion in general. The question about compensation. Do you hear me?
Yeah.
Yeah, about compensation. So part of the ambiguity is that when you see partial regain of the functions, there are two ways to interpret it. One, to say that there is some plasticity such that other regions take over these functions, or you can say that the problem was the trauma of the lesion itself, rather than the fact that you don't have that there is some tissues which are missing. It's not the missing of the tissues which caused this transient loss of behavior or change of behavior, but actually the trauma of the event itself that caused disruption of function. So this is part of the ambiguity in interpretations of lesion studies.
Mm-hmm. Okay. Any other comments? Okay, so now let's look into another type of disturbance or perturbation that we do to the system, and that's by brain stimulation. So this is active. I mean, we plan it and we stimulate the brain, and that's how we get some manipulation and perturbation. And let's look at one example, which isn't it's a classical one, but it's a nice one, and that is an experiment. I mean, this was done with electrodes in human brain, which of course cannot be done in regular conditions. So this is done because in preparation for epilepsy operation, and usually the operation is taking out the focal point of the epilepsy, and in order to know where the focal area is, so you have electrodes in the brain which record brain activity, and then when for like a week or two, and then you know where the area is, and then you could do the operation. However, while they're waiting for the operation, you can do experiments on these patients, and as you see here, you also stimulate areas in order to know exactly where the electrodes are. So this experiment was done by Yitzhak Fried, and he found that if you stimulate the pre-SMA, which I show here where that is, next to the motor area, at low current, that elicits an experience of an urge to move a specific body part. Okay, and if you give a more intense stimulation, then it often produces a movement by itself of that body part. So what you see here is something which we will talk more later when we get to talk about the free will and Libet experiments. And that is the notion of some experience you have of an urge to do something, an urge to move, an urge to act. And this is a conscious experience. And what he shows here is that by stimulating that area, he got he created just the urge, nothing moved, just the urge. If he stimulated stronger, then the movement itself was created. So this is one example of a brain stimulation. So it's as if, in a way, a normal cycle of the brain of movement is like the regular cycle. You interfere and you put a pin in a middle point, and you start the cycle from that point. So instead of the whole intention build up, which ends in an urge to move and finally ends in movement, here we put an electrode and we just started the process from the middle, and then we started the end part of the urge to move and then movement. So just so it's another in a way a causal element which we put in the middle. Another example is with without opening the skull, is a TMS, which is transcranial magnetic stimulation. It was mentioned in the first lecture for a moment, but what we have here are currents that electrical currents going in this kind of a spiral, and then it creates a magnetic field, which in turn creates again an electrical field in a certain location in the brain. So it's a way to stimulate electrically the brain from outside. And the experiment I'll show you now has to do with a game called the ultimatum game. So the ultimatum game is the following game: is you have two participants. One offer, and these two participants, one gets, they have a certain sum of money, let's say $100, and one, let's say the boy here has to offer the girl here has to offer, how much is he willing to give her from that $100. He could offer $50. In that case, it will be a fair division. Or he could say, tell her, look, I'll give you $5, and then it will be, and he'll get the $95. And now she has to decide if she agrees to this or not. If she agrees, she gets what he offered. If she doesn't agree, then they get nothing. He doesn't get, and she doesn't get, and it's and then this trial is canceled. Okay, I mean they get no money. Okay, so now you have what do we have here? We have actually two sides to this. On the one side, you have economic self-interest. I mean, if he offers her $5, economically, she should take it. I mean, the other option, if she says that she's not willing, is that no one gets anything. But because it's not fair the way it's done, so many times people will react and say, no, we're not willing. I don't get and you don't get, and forget it. It's not fair. They're looking for the fairness. So it's either you take the money and then you're interested in you have economic self-interest, or you say no, and then because it's not fair. So now they did this experiment while giving a TMS while activating the TMS. And the interesting results were the following. They activated the TMS in the dorsolateral prefrontal cortex, and on the right side and on the left side. And what you do with TMS also as a control is you do as if it's activated, but it's a sham. It's a sham trial. I mean, nothing's being activated. It's like a placebo in medicine. I mean, you just do as if you're activating it, and you don't do anything. And now you see how people reacted. And what they noticed in this paper is that when activating the right side of the dorsolateral prefrontal cortex, what they got was that there was more acceptance rate of the offers. That means that if I was offered $5, usually the normal, again, I'm not saying if it's good or not, but the normal reaction would be, no, it's not fair, forget it. I won't get and you won't get. And while stimulating the right side of the dorsolateral prefrontal cortex, people didn't care so much about the fairness, and they were interested in the money and the economic self-interest. So this is interesting, because you hear this, you say, okay, there's some connection here between that area and a measure of fairness or economic self-interest, but you need a larger theory to decide what is going on here. What is the normal thing, that is the normal thing to go for your self-interest, and the dorsolateral prefrontal cortex usually inhibits that, and that's why you go into fairness. Or is the normal reaction, no, it's not fair, and then I won't take it, and then you have to inhibit that in order to have economic self-interest. So the question is, what is the baseline here, and what reacts? But this experiment could help decipher between these options. So to sum up this case, so applying low frequency repetitive TMS to the right, but not to the left, dorsolateral prefrontal cortex, increases economic self-interest and reduces subjects' willingness to reject unfair offers. Okay, so these were two cases of stimulation, one with electrodes and one with TMS. And if we look at these cases, we first of all, they're short-term effects, not like lesions, which are of course long-term. If we talk about electrodes, so they're invasive, they damage tissue, and they usually are not applied to humans except for medical procedures. And TMS, on the other hand, is more difficult to stimulate deep brain areas. And it's also the question is the focal point, how much could you focus with these measures when you try to stimulate? Are you stimulating the right area or not? But finally, it seems that you do have here a causal role. And again, the interpretation is not always clear, because the question is okay, I know it's part of some causal chain here, but is it the immediate chain? Is it part of something else that causes it? And also, as I mentioned before, the issue is, in order to say something causal, you need a greater theory of what part. How do you look at it? And like we showed here, how do you understand at all what happens in this ultimate game? It's not clear what. I mean, I oppose two notions of self-interest versus fairness. But are these notions, what does it mean? What's the baseline? What is the other side of this notion? Okay, so this is what these were brain stimulations, and these two types of perturbations were brain stimulation and damage that you have to the brain. Yeah.
So allow me two brief comments. One is that anatomical, that when you stimulate a region, human brain, that you cannot elaborate more on the specificity, you may actually stimulate axons, processes that go through this region rather than neurons that are in this region. So actually, you may actually stimulate the activity of nerve cells which are not here, which are far away, because you stimulate their axons. That's one complication. The other one is kind of elaboration on what you said, but also what I said in the first lecture, that in general, in any population of neurons, in any local circuit, there's excitation and inhibition, and when you stimulate, you either activate more this region, and then you generate whatever behavioral consequence, or conversely, you actually amplify the inhibitory component of this region, and by suppressing activity, you allow other processes to express this behavior. So that's part of the ambiguity, that you really have to understand better what is being activated in the circuit in order to interpret it.
Yeah. Any more questions? Okay, so let's move now from the side in which we control or try to, like we said, perturb, to observations and measurements. Okay, now we're going to look at the brain and try to see what we could say about the brain in these cases. And we'll do this through three basic examples. One example is a single unit measurement in monkeys, which will be connected to decision making. The other example, we'll use EEG, and another method, we will use fMRI. We will look in an fMRI study. All these cases are, we'll try to understand to get a feel of how this connection between these high cognitive notions to actual brain activity. How are they done? So we'll start with the decision making, the monkey. So we have here the experimental paradigm is as follows. You have a monkey sitting in front of a screen, and you present to him two targets on the left and on the right. And then what starts happening is there is what's called random dot motion, is you have dots on the screen which are randomly moving from to the left or to the side, to the right and to the left, and if it's random, you would think at some moment it might be moving to the right, at some other moment you would think it's moving to the left, and at some point, the monkey, when he decides the direction of movement, he does a saccade with his eyes to the target, either on the right or on the left. And you do all kinds of measurements here. One is the reaction time. the time from the moment which the random dot motion appeared on the screen until the moment he does the saccade. You measure, and here you also have measurements of the brain of the monkey. The manipulation that's done here is that you vary the percentage of the coherently moving dots. I mean, you could decide how many dots, what will be their coherence? I mean, will it be or how many dots are, let's say it this way. If it's 50-50, then there's no direction there's no net direction to these moving dots. However, if they will all move in one direction, then it will be 100% coherent in one direction. And you could vary the coherence in all the middle and all the possibilities in the middle, and you check the monkey's reaction according to these different percentages of coherency. Is that clear, the paradigm? Yeah, okay. So it's a simple paradigm, but it has this important element of coherency, which we can make use of. So first, let's look at the behavioral results. So the behavioral results are as we would expect. If we look on the bottom figure, so what we see that the coherence, that's the x-axis of the motion strength, is the percentage of coherence. The higher it becomes, that means it's more coherent. That means the monkey reacts faster to the task. I mean, he does the saccade faster than the cases in which the coherence is low. Okay, and also the opposite side of this is how many times was he correct or made a mistake. The more coherence there is, the more correct he is in his response, and the less coherent the dots move, so he makes errors. So now let's look into the brain activity. So the brain activity we're looking at is the area that's involved in the selection and preparation of the eye movement. Okay, remember that's the final moment where he moves the eye, does the saccade, which is in the lateral intraparietal area. So we're looking at electrodes there, and what we see here, look at the curves which going up. What we see here is the blue, red, and orange or yellow curves are actually the difference in the motion strength. The blue is the least motion strength, and the yellow is the most strength of this coherence. And what we are measuring here, the y-axis is the firing rate, I mean spikes per second, and the x-axis is times. What we see here is actually the stronger the motion strength is, okay, of the dots, the faster you the slope is stronger. I mean, the slope goes up faster, and that makes a lot of sense. I mean, the dots are more coherent, and then the firing rate goes up faster. Now, you should notice that what we see here are averages over trials, over many trials of the firing rate. Now, when we do average, we need to lock the average onto something, to a certain moment. So here we're locking all the average to when the Q appeared, when the dots appeared on the screen. However, if we lock the average to the sakad, to the moment of the movement, what we see is this. What we see, these are the same lines we saw on the left. However, this this line is the eye movement, and we do average to the eye movement. And what we see, this is the end part of the whole story. And what we see is that the ramping up of activity reaches the same point before the eye moves, and then it falls back to baseline. So what we notice here is that it doesn't matter what was the motion strength of the dots. The moment of the movement of the eye movement happens only when the ramping up of the firing rate reaches a certain threshold. And here you see nicely that they all reach the threshold of about 68 spikes per second. And when that happens, that is the indication or the point. Soon we'll see a model regarding this, but the point in which the instruction to the eye to move comes. Okay, before I move on, are the results clear here?
Ariel, can I ask a question?
Yeah.
Why is it a model of decision making? Because the monkey doesn't like compare two rewards or something like this. It has it's completing a task more than doing a decision, or what we will call a decision between two different things that it needs to compare independently of the environment.
Look, here the decision is a perceptual decision. I mean, he has to. It's not a decision what to do. He's trained to do the sakad in the direction where the motion is, and we're trying to. I mean, what you're saying is a good point, but the different types of decisions, and the decision here is perceptual. We're trying to see when he decides that he saw the direction, or when he decides that the direction is to the left or to the right, and that's why it's not the decision making which you would say. Let's say you have to decide between the different options with different rewards or things like that. Okay. Any other questions? Okay, so now, given these observations, what was proposed is a model which is called drift diffusion or accumulation to bound model, and this is a model in which what we could observe here is that what we see here, this curve. Let's explain what we see here. The y-axis. The y-axis is the amount of accumulated evidence, and the x-axis is time. And what we see here is actually the integration of the evidence. Now, what do I mean evidence? Evidence, I mean the sensory input that came in. It comes in as evidence because he needs to decide, and we're talking about perceptual decision making. So there's evidence coming in, which is the sensory input of the dots, and you have to decide where does this evidence lead you to. So what we see here is a accumulation of the evidence, integration of it. And sometimes you could see here, if you look at the figure A, you could see it seems clear that it's going in one direction, and sometimes it's going in the other direction. However, the mean slope of this is actually correlated to the motion strength of the dots, of the evidence itself. So the evidence goes and accumulates, and when it reaches a threshold, that's the moment in which the instruction goes on to the eye to do a saccade in this direction. However, if the evidence would go downward, these are two different thresholds, plus A and minus A. If they would go downward, and then it would actually choose the other saccade direction. And let's say the coherence of the motion would be zero coherence. I mean, it'd be totally random. So what you would have is actually a movement around the center here of the figure. However, it accumulates sometimes in one direction and sometimes in another direction according to the noisy input that it receives. And if by chance it would cross the threshold, then there would be a movement even in the case of zero coherence. So this is basically the model of decision making using a drift diffusion model. That's the general idea, and what you see here is a kind of a mechanism of neurons that accumulate. Their task is to accumulate evidence, and when it gets to a certain bound, is to send the evidence onward. It is to give the instruction onward to do the movement. Of course, when you see such a model, a few questions are raised. One of them is, who decides where this threshold is? What does it mean to have a threshold biologically? I mean, how, and to control it, to decide where is it considered enough evidence for that crossing of the threshold will be the moment that the saccade happens. And another issue is, of course, when you see such a model, is to ask yourself, okay, what area in the brain, or what kind of areas could have this type of accumulators which might fit this this story. Yeah, any questions? Okay, so I just want to sum up this this example. So what we see here are the questions. In a way, we're asking more questions than answers, but I'm trying to do it through the examples. Is what can we learn from the observations? I mean, we've had an observations, and we looked at these ramping in a certain area, and then we had these saccades. Could we infer any causation, or is this only correlation what we see here? I would be happy if you have any input on this. I would be happy to hear what you have to say about these. How do you see this example? Do you feel that it's a causational example, or is it only, did we give here only a correlation to some behavior that we had here?
Well, the question, what was the ultimate causation, like on a time span when, of course, if something precedes something else, we can say it's part of the causal chain. But where the ultimate, like, first chain starts, that's the different question. I mean, if we ask about causation, we're always talking about causation chains. If something precedes temporal something else, I think it makes sense to say that there's some kind of causation. It's part of the causation chain.
Yeah. So, but here, that's interesting. So, but you could say the beginning comes from the outer world. I mean, it started from the stimuli, and then it went into this, it got into this area which accumulates, and then you have a movement. And you could draw the whole circle of causal relations, and that would be a causational explanation.
But what if my brain connection, I don't know, or the monkey's brain connection is like shifted, that it always prefers a little bit more to the right? Like it's, I don't know, the circuitry is biased towards one of the choices. That will be also part of the causation chain, which happened even before you give the stimulus.
I guess you're right. Yeah. Mm-hmm.
Yeah, you can say that there are structural causes that generate some bias embedded into the structure of the circuit, functional structure. Absolutely.
Yeah.
But I think that it's an important question, but correlation versus causation. But it's a question which permits science in general, science discovery, that not always, or it's mostly about measurements and observations. So it's a broader scientific question. How do you discover causal relations or natural laws from observation? So of course, in the brain everything is more complex, but yeah, I think there is a more general question about scientific discoveries in general.
Yeah. No, I agree with that. Coming from the previous classes, which we talked about physics, and there it's easier to see when you talk about causation. Is it causal or not causal, or when the systems become complex, so then you ask them, so what is the science? What is science are we trying to do here? We have observations.
There is also a question of resolution with whether the brain part, even if we agree that in this experiment there was some causation, some sort of the causation, you need to discuss whether it was the brain part that caused this behavior, or the single neuron, or maybe even the single synapse, or even a chemical in the synapse. So this resolution are not equal. You can't say if you stimulate the brain region and it caused some kind of behavior, and even if we agree on the assumption that it is some kind of causation, it can be only a single neuron or a single synapse in this brain area that actually responsible, and all the other are just something else.
Yeah, even though, in this story you have a mechanism, and the mechanism uses more than just a specific chemical. I could say that sometimes this chemical causes one thing and not the other, but when you try to draw a mechanism like we tried here actually with this accumulator, so it's a specific mechanism. So you would say, okay, these things do have a resolution, which are the building blocks of this, but the level of resolution which we're interested here is the neuron, and etc. And but you're definitely right, because these little things can change, of course, the outcome.
Because you don't have the whole chain of causation, I think it's problematic even to say that it's a neuron, because you see a neuron, you say behavior, but there is a lot of parts in between that are missing, and then even to infer that this is the neuron caused it, it's problematic if you don't know what the effect of the neuron. It can be only a second synapse in the neuron, and or even something else.
Yeah.
But I think the mathematical model here kind of helps us at least to rule out something. So the bounded diffusion model, on one hand side, you can say it's a model of the behavior or decision making like behavior, and you can test it on purely prediction about the behavior. On the other hand, the way I think Ariel presented it, it's more than that. It is a model that it is a proposal that already a single neuron represents or computes this kind of an accumulation evidence, and that is suggested by the experimental results about single neuron firing rate. They seem to follow the trajectory of the noisy evidence and reaching a bound. So then this experiment suggests not only that the behavioral level is bounded diffusion, but also that the single neurons seem to execute this type of computation. Now, you're right that it doesn't mean that the single neurons are the decausal. It may be something else. You need a perturbation to do this, but that's the claim that you already see it at the level of single neurons. It's like tuning curves, and that I described in the first lecture, that there are some computations that you can read off the activity or the modulation of the activity at the level of single neurons. But what you add in, but it is true that, again, it's a theme that we will come back to it later on in the course, that in complex systems like in the brain, it's always what Ariel called mechanism. It's always, one can talk about levels. You can ask how this mechanism reflects or explains the activity of single neurons, and then you can ask, at a lower level, how synapses, what is the mechanism that generate this signal in the single neuron? So then you go to the synapses level, and maybe even to lower level or to higher level about the whole area or population of neurons. So I think typically mechanistic explanation in the brain will typically involve this kind of multiple level description.
Yeah. So now I want to, one more point, maybe before we go to a break, is that, is the second point here, is that we have to be cautious in moving from observation to interpretation. I mean, we did here an elegant move. I mean, as Haim said, we saw the ramping, and then we said here, this is the model, which seems to follow nicely this type of ramping. However, we should notice that that's not always clear, and in this case specifically, there were criticisms about seeing the ramping, and this came out of a paper from 2019, seeing the neurons as doing this ramping that we saw, since what we observed wasn't the ramping of a single neuron, but was a ramping of an average of trials, and averaging over trials. That's what I showed you basically in the figure before. And what they try to show here in this paper is that even if the neurons are not ramping, but are actually doing a step function from no activity to full activity, also those, if you take averaging over trials, what you'll get is a ramping like activity. I mean, and this isn't so in a way this ramping doesn't have to reflect what is actually happening. So you have to make sure when you look at you observe something, you have to ask yourself, what did you observe? How do I interpret it? Am I observing the thing itself, or am I observing some mathematical, let's say average in this case, which created this observation? And that will be reflected also later on when we talk about free will and about Liebet's experiments. Okay, I think I'll start. So our next example will be an example with EEG about error and conflict. Now, before I start the specific experiment, I want to share with you something that you should be aware of, is that different imaging techniques have different spatial and temporal properties. So if you look at the figure here, what you could see is that EEG and MEG have good time resolution. They can measure milliseconds, and that's their accuracy, as opposed to fMRI, which is mainly good for a few for experiments where there's where the speed of activity is in seconds at least. So on the other hand, of course, the spatial resolution is better with MRI than with EEG. The EEG is on top of the skull, and it measures the electric activity, electrical potentials from here, but it doesn't. So you could infer what it's measuring, what areas of the brain are underneath every location of EEG electrodes, but it's not such a good resolution spatially as compared to MRI. Okay, so let's go into the experiment I want to show you. This experimental paradigm is called the flanker task. It's a very simple task. I'm trying to show simple things so it's easy to get a grip of them. So the task is the following. What we have here are we have a participant sitting in front of a screen, and on the screen he sees arrows. So the arrows could be like the top row, in which all the arrows are in the same direction, and what he has to notice and he has to press accordingly, press with his left or right hand accordingly, is the middle, the central arrow. He has to press the direction of the central arrow. So the central arrow here is left. He has to press with his left hand according to this central arrow. However, the central arrow could be in the opposite direction of the other arrows. So the central arrow is the cue, is the target that the participant has to notice, and the other arrows are called the flankers. This tried to interrupt either interrupt or go in the same direction of the target arrow. Of course, everything could be in all directions. In this example, it's to the left. Of course, it could be to the right, and the other way around. So these are two possible trials. Either you get a congruent trial or an incongruent trial. There's a third possible trial that you could get, is a neutral type trial, in which you have the central arrow, but the flankers have no meaning. They have no direction to them. So when you check the behavior of such a task of pressing left and right according to the central arrow, and you have to press fast. That's also part of the idea. Is you get the following results. Let's look at the results. The red in the middle is the incongruent cases, the case in which the arrow that you have to press accordingly is in the opposite direction to the flankers. The blue is the congruent cases, and the gray is the neutral cases. So first of all, if you look at the left figure, you notice that the reaction time, the time that it takes from the moment you see the arrows to the moment you press, it takes more time to do it, to press, if the arrows are incongruent, if they are in opposite directions. And that makes sense in a way, because you have here stimuli which is interrupting, and you're trying to press according to the central arrow. And if they are congruent, it's faster, and you see that the neutral is close to the is somewhere in the middle or closer to the congruent case. Also notice that you have more mistakes, more errors in the case where the incongruent case. I mean, in the incongruent case, you see you have 6% of the trials are errors as opposed to the congruent or neutral case. Okay, so this is the task, and one of the issues that comes out here is what happens in these error cases, or what happens as a result of the error case. There's a lot of issues here that I'm not going to talk about. I mean, what is the process of again, like we saw before, of evidence accumulation, or of the conflict there is here to decide what to press. But I want to focus on one little element here, which is the element of the errors. So as you see, in the incongruent cases, there are many more errors. Also, but you also have errors in the congruent case. And now there is an interesting measure which is called the error-related negativity, which has to do with a brain signal after you have done an error. Okay, so you're doing the task. You want to press. You see the arrow is to the left, but by mistake you pressed with the right hand, and you have made an error. So let's look a little into the error-related negativity, which is measured with EEG. So when you have EEG, okay, here's a participant with an EEG cap, which has electrodes. And what you do is you measure the electrical potential in a certain point here on top of about the area of about the motor area, or a little frontal from the motor area of the brain. Now, in general, I'll just explain when you want to have such a one of the ways of measuring EEG is to look at many trials in which, let's say in this case, you had errors. Look at the brain activity in those trials and average over all that brain activity at those trials. When you lock the average to, in our case, to the response. Why do you lock it to the response? Because the response was the moment where you did the error. So you did an error, and then there's some brain activity which we're interested in, and you lock you average over all the trials at that case. And why do you have to average over all the trials? The reason is simple. since the EEG has a lot of noise in it, okay? However, the noise is not correlated. And if we average over many trials, what will happen is that the noise will cancel itself. And what we'll get is only the brain activity, which we are interested in. So here, we're interested in what happens in the brain after someone did an error. So again, we take all the trials where we did an error and we measure this activity. And what we see is the following. Okay, so let's look at the y-axis is in microvolts. This is a schematic. It's not a real results, but it's taken from a review on the issue of error-related negativity. And what we see, let's focus on the red and on the black curves here. So what we see is the red curve is an average locked to the button press of an error, is the cases where you did an error. And the black is the cases in which you were correct and you didn't do an error. Okay, so we have the two cases, an error and a non-error. And what we see is a clear difference in the signal. What we see is a bigger negativity, it's called a dip of the red signal after there has been an error. And the black has a small dip. So it's not so it's called the correct related negativity, which is small negativity, but the red is an error related negativity, which is larger. So here we have an index, kind of an index to errors, because we know that what happens is after you press and you have an error, this is what you see. However, and this is I like to call this error-related negativity the oops signal. After you did an error, I'm sure you know, and you do something fast and you press something wrong, you said oops, and that's exactly what you see in the brain, and it appears about 150 milliseconds after the button press. And the question is, what is this error-related negativity? And it's interesting to see the history of this error-related negativity. I mean, what does it index actually? And here we're going back to our question. What are we doing when we do this type of correlation of, okay, there was an error, and here we saw some activity in the brain, which is nicely in many studies, you could see it repeatedly again and again. It's not something that you found once. I mean, there's good science here, but what is the science telling us at all? And so there's an ongoing debate. What does it index? So here I'm just showing you. So it really started in the 1990s, as an error detection signal. It's clear that has to do with error. Then they came later on, about 10 years later. Wait, what does error mean at all? Maybe we could say something more general. Maybe it's not an error, maybe it's conflict. Okay, so it's actually the detection of conflict, not conflict in the decision making, but conflicts that happens right after you made an error. You actually have two conflicting aspects in you. You have a kind of the reply that you did, and you had what was right to do, and you have a conflict between them. Maybe it's defining actually a conflict and not an exactly error. And then you could see on as the years went by, they had other proposals, prediction of error likelihood, and a more general kind of signal of action monitoring. Okay, the monitors, we have a system that monitors our actions, that actually looks into what we're doing and all the time thinks, is this the right thing, or is this should be done, or etc. etc. And also issues came up. Do we have to be conscious or not conscious of this error? I mean, what is the meaning? I mean, does this correlate with us being with this error happening only when we are conscious do we see this, or also when we're non-conscious do we see this? And also the observations here are not so clear, and there's a debate regarding these issues. So as you see here, you see a certain observation, but the interpretation, or how to see how large of a function to relate to it. Is it only error, or is it conflict, or etc.? I just want to show you here results that we have, that we in the experiment we did, which has to do with free choice picking. I won't go deeply into this issue. I just will mention that free choice picking is the case in which you have two options, and you have to pick between these options, but these options are there's no preferable option. You just need to pick one of them. I mean, you don't need there's no significant meaning to your picking. It's a specific one possibility over the other. They're both good options, and you just have to pick, as many people in the supermarket are standing in front of identical items, and you just have to pick one of them. And so, you do the picking of one item. So in an experiment we did, I won't go into the experiment itself, but it has an aspect of this free choice picking. And what you notice in this experiment, in certain cases, in certain trials, is that sometimes there's this kind of a deliberation between the options, although their options are equal, and it doesn't matter what you do, there is a kind of deliberation between these options. And you can see it's deliberation in many, I call it deliberation, but it doesn't have to be deep. I'm just saying that you intend one thing, and then you change your intention to do the other thing. And so there is kind of going back and forth between the options, although the options are both equally same for you. And one way of measuring this is through electrodes on the muscles, and you can see that even the muscles are activated sometimes when you intend to do one thing, and the muscles are activated. However, it's not executed, that action, and then the other muscle is activated and executed. And this is what you see on the bottom figure, is the muscle activity. The red is one hand, the black is the other hand. So the red had muscle activity without, let's say you have to pick which button you're pressing, and it doesn't matter which you're pressing, left or right. And the left hand, you could see muscle activity but no button pressing, and then the right hand has muscle activity and there is pressing. Okay, so this is a kind of what we call change of intention or deliberation between the possibilities. And now when we look at this case, it's interesting to see that when someone changes from without executing it, but changes from one intention to the other intention, there also is a error-related negativity. I mean, this negativity that we saw before that's related to the errors, now that's the red figure here, is the case in which someone just changed his mind between doing option A and doing option B. And then here you see there's also the same error-related negativity. So I think here we see a clear answer to the question, is error-related negativity related only to errors? Of course it isn't. I mean, here there's no error because there's no right and wrong here of what you do. You can do whatever you want. and even though there's no error, there still is an error-related negativity. But if going back to our question, it's interesting to see that you have these signals in the brain, and the interpretation of what they mean or to what concept to connect them, that's the big question. And here, if to sum up this thing, is the question is the way we parse our mental or psychological concepts the same as the individuation of brain mechanisms? Who said that the world, in the way we divided the world into notions of error, this is conflict, this is the deliberation, this is such and such, etc. We divide the world, and we see someone behave in a certain way and another person behave another way, and we give names to these things. And these could be names that appear only that appear in the natural language or in psychological language. But all these names that we give to things around us, who said that they have to map in the same way, in the same individuation into the brain mechanisms. And the follow-up question is: should we expect that our natural language map nicely into the neural realm? And if it does map, in what resolution? I mean, what are we looking at it all? So the whole project, in a way, raises a question: what are we looking for when we're doing this? I started with all these notions of high cognitive words, and they said, I asked, how do we map them to the brain? But the question is, what are we mapping? Like, why should the brain be mapped accordingly to these words? And a nice, here's a reference to such a critique you could see with a book by Buzaki. We'll put maybe a paper of his and reply to this paper in the website. And you could read for yourself the kind of critique that he has here. But I'll just say this. Okay, I'll stop here. Are there any questions?
The signals that ERN, what is it exactly? Where do you record it from?
So, if you could see here.
Yes.
Here, this is, let's say, a brain. So I recorded from the top of this area. I mean,
so you don't know exactly what creates it. It's just a marker that you see. You don't really know where it is.
We know it's probably it's over the area the ACC, what's called, and which also is involved in conflict resolution and issues like that, and they think that it might be there. But the question is, what part does it take in there? Is it a result? Is it something for? Is it a preparation for something else? What is it exactly? All that is under debate.
Okay, but resolution-wise, it's not like in specific ROI or whatever. It's something that a marker that you see, like, I don't know, for example, I don't know.
Yeah, no, here, as I said, the resolution is, with the EEG, we get the resolution.
Yeah, but can you map it to a specific area or not?
So they, putting together other experiments, they say it's the ACC, really.
Okay, got it. Thanks.
Yeah.
I have one question. So.
Yeah.
Let's say if the subject is given enough time to choose for the options, how would it affect the ERN activity? Like, would you see the similar kind of error-related negativity, or would it change?
These negativities are basically in rapid, fast reaction time experiments.
Okay, so because I'm asking if
you give time and someone replies, they usually, that's why I put it together with the oops. Usually the oops comes in this type of faster reaction time experiment.
Okay,
I have a question.
Yeah, who's asking?
I mean, it seems that it is assumed that physical activities and mental activities map to each other on a one-to-one basis. But wouldn't it be reasonable to assume that many different mental phenomena can be mapped to a single physical phenomenon or vice versa? For instance, what you call error-related negativity, I mean, maybe it is mapped to errors, but also to different phenomena. I mean, I don't see a problem with that.
That's a very good question, and that's a central issue in this mapping project. Is really what is these relations? Could we talk about one? I mean, one brain activity that maps different notions. Now, in a way, the notion that I showed you here is might be embedded in each other. So it's not different. So different. I mean, you could say error is part of conflict. It's not different activities. But let's say they were different activities, or different different human functions or activities. And so you would have the brain activity mapping these two different things. So what would happen when the brain is active? What are we experiencing? A or B? I mean, if they're two different things, what are you? I mean, you would expect that there would be some kind of correlation that will decipher between what's happening now. Am I experiencing A or am I experiencing B when this thing is active? Okay, so it won't be clear what's happening if you really take into account that the brain has to be active when you're experiencing things. Is that clear?
I'm not sure I understood the question. So, why Yishai? What do you mean by many to one? Well, in one direction I can understand, but you think that the same brain activity can be mapped onto multiple cognitive states or processes?
Yeah, I think. I mean, I don't see why not. I mean, as to differentiating them, I mean, it's a different question how it is differentiated, but I mean, it could be in a different part of the brain, I don't know, or something like a higher function. But I don't think it's necessarily something that's, illogical or something we should rule out. It seems to me, I know.
Well, it's an interesting point. I mean, I don't know if it's logical, but you have then to tell us an explanation. You owe us an explanation. How come the brain activity was the same, but the outcome in terms of behavior, or let's call it behavior, was different? So you will have to say, well, there is something in the environment or something in the body. You need to explain, right? It's not so simple.
Understood.
But I'm not saying it's impossible, but you will then have to come up with a theory of cognition which goes beyond the brain, which that somehow the same brain activity may generate different cognitive states depending on something else. So what will be that something else? You will have to tell us. Okay. But on the other hand, in the other direction, I think it is definitely more plausible that there are multiple different brain activities which might yield the same cognitive state or the same even action. And that's, I think, is we will discuss this later on, but I don't know if Ariel wants to comment more about that.
Yeah, I thought of leaving it to the end, and I'll just say, and then I'll
so we'll come back.
about
We'll come back
what's
to
called
that.
multiple realizability.
Okay.
This option. Okay, so we saw this. Okay, I want to go to our final example. I see we don't have much time, so I'll try to do it. Our final example has to do with conscious perception using a method called binocular rivalry. And then here we will use our measuring tool will be the fMRI. So follow me with what we see. So subjects, first of all, view a sequence of alternating stimuli, okay? Which the alternating stimuli is a house, a face, a house again, a face. Okay, I saw it flashing. I don't know if you see it. Did you see it through the Zoom?
Yeah.
Yeah. Okay, so it flashes a house, a face, a house, a face. Only these two stimuli, one is a face and one is a house. And what we do when you're in the MRI machine is we try to map the brain activity and see what is active, what brain area is active while showing a house, and what brain area is active while showing a face. And then we discover that there are two areas, main areas. One is the face area with the fusiform face area, and the other the parahippocampal place area. Okay, so we have these two areas which are active. And now let's see let's follow the activation. We show at first nothing, and then we show a face, and then we see that at the face, when we show the face area of the brain is more active in the MRI signal. And then we show nothing, and then we show a house, and then we see the red is the house area is more active, and so on and so forth. We go on showing a face and a house, and we could see nicely that each area is activated when it perceives the different stimuli, a house or a face, accordingly. Okay, so this is the basics. Now, what we will do is that we create a binocular rivalry stimulus. So I don't know if you're familiar with this notion. The binocular rivalry stimulus is such that each eye sees a different stimulus. Okay, the left eye will show it a face, and the right eye will show a house. How do you do it? One of the ways to do it is the way you see here. You put these glasses with red and green colors, and then you put actually the two pictures one on top of each other, and each eye will see the picture which it could see through the color it has. So one eye again sees the face, the other eye sees a house. Now, you might think that when you see such a thing, you'll see a combined picture, like what we see on the bottom. You'll perceive a combined picture. But actually what happens, and you should try it at home, like you say, what actually happens is that you see one picture, and that picture fades out, and then you see the other picture. So you see a house, and then that fades out, and you see a person, and then that fades out, and you see a house. That goes and alternates every few seconds. It alternates, and you really experience seeing a house or a person, and it changes. This experience changes while you are doing this. And so the task is that the subjects have to report what they see. Do they see a face or a house? So you're looking at this, one eye has a face, one eye has a house, and you're saying, now I'm seeing a house, now it's fading out, I'm seeing a face, fading out, I'm seeing a house. And what's interesting is that if we, looking at the brain measurements at the MRI results, what we see is that also when people, I mean, like the case when, the bottom case is the case where the pictures actually change in front of you. And we saw that once the face place was stronger and once the house place was stronger, the signal was stronger. Also in the case where the percept is fixed and each eye is seeing the same thing, nothing's changing. I mean, the percept is the same, the left eye is seeing a house, the right eye is seeing a face. Also then, there is a change in the MRI according to what you're perceiving. So realize here that there's no change in the stimulus, there's only a change in the perception. And we're measuring here the perception. So we're measuring the conscious perception of seeing a house and then the conscious perception of seeing a face, while nothing is changing in the stimulus. Is this clear? Okay, so if you understood the experiment, so first of all, it's a nice experiment to do with conscious perception. And the question is, what could we say about what we just observed? I mean, we observed something, which is nice to see what we observed, but what does it mean? So this is, first of all, it goes into, but I don't have time to get into it, into the larger context of what's called neural correlates of consciousness, NCC, and that's has to do with the science of consciousness, is looking in the brain what areas does consciousness hide? I mean, and what areas are important for having a conscious perception. So here we saw two areas, which, yeah, I mean, measuring them, we saw that they measure the conscious perception. So are these the areas that consciousness lives in? Of course, we wouldn't say such a thing because it's, of course, task related. Here we showed houses and faces. It shows something else, so it would be something else. And the other thing is, what kind of, what are we looking for? I'll just leave it as a question. What are we looking for when we look for these neural correlates of consciousness? As it implies by the name, you're looking for correlations. You're looking for correlates of consciousness. Could we give anything causal, any causal interpretation to consciousness? So now that brings us to a very big issue that if you look at the concepts I put in the beginning, so consciousness is somewhere in the middle, is very central to human cognitive states. Okay, so to sum up, what we saw today is, first of all, that lesion studies and perturbations, those were the first two types of studies I tried to present in front of you. Stimulations, they could infer a causal framework. However, we need to be careful in the interpretation. Well, how causal are they? What do they mean in their causal, in this causal framework? Observations on the other side, first of all, definitely create a correlational framework. The question is, after observing something and creating and defining a mechanism, do we actually have here something that's more on the causal side? And why are we asking all this goes back to what Haim said in the beginning, which is part of a general science question of what do you do when you do this type of science. And when we show these mechanisms, the question is how they are physically realized. And talking about that, we come to this question which was raised, which came up before, is regarding the functions that we see and how are they realized in multiple in multiple ways. Could one function be realized in different ways? Could they be realized with neurons and also be realized with electricity? I mean, in some kind of machine, or it could be realized in humans and in a monkey. I mean, this aspect of multiple realizability isn't definitely a problem that a certain function could be realized or in different ways, but still express the same function. But it raises the question: what kind of science are we doing? If one function could be realized in different ways, so what are we doing next? Do we compare the ways that it's realized, and we find the underlying thing that connects all these things that's why this function is functioning? And if we do that, so basically we're going back to not multiple realizability, but to one aspect which realizes the function. So this is a central question, which today I'm just raising it, and I hope when we go on, we'll have more time to discuss it. So I'll stop here. Time is up.
Before time is late, so before we depart for today, for this week, I just want to remind you that next week we're going back to physics, in this case thermodynamics and statistical mechanics. These are exciting, interesting, and fundamental issues that we need to absorb in order to continue later on in our journey. Those of you that are not familiar with the foundations, with the fundamental laws of thermodynamics, etc., or with the concepts, I urge you to schedule a tutorial hour with Ariel. He will go over some of these introductory materials. We'll also post background literature for those of you that are not familiar with the basics of thermodynamics.